back to my channel. So today we will talk about the book that I read by Philip Detmer called Immune. So it teaches about the immune system. But what is the immune system? Immune system is a system that helps you from the other invaders that wants to invade your own body. They want to do this because they don't have to do any work to get their nutrition. So they want to be inside your body. So the body developed a way to stop this by the immune system. There are two types of immune system: innate immune system and the adaptive immune system. Innate immune system is the immune system which acts first, like the dendritic cells, macrophages, neutrophils, and the complement system. The adaptive immune system is when the dendritic cells take samples from the bacteria, and then they activate special cells which then release antibodies and more. We will talk about this book in this video. So, first things first, this book talks about many things, about different kinds of proteins. About many different kinds of proteins. There are many proteins released by the immune system. Here is a diagram given about the things in the immune system. Now there is an interesting diagram here about the different kinds of bacteria and their sizes. These are neutrophils, and these big things are macrophages. This green thing is the E. coli, and this is the red blood cell. The neutrophil is 10 micrometers, and the macrophage is like 21 micrometers, and the red blood cell is 7.5 micrometers. Macrophages are twice as bigger as regular cells. Now there is a neat diagram given here about the E. coli bacterium. Like it is said about the flagellum, about the plasmids and the DNA, the plasma membrane, the cytoplasm, the cell wall, and the capsule. Then it talks about the first layer of defense, the skin. How does the skin protect against the bacteria from entering? First things first, if a bacteria lands on your skin, first things first, there are a lot of, the sweat has lot of salt in it, so it is very saline, and the surface of your skin is acidic. Plus, to stop the bacteria getting feeding onto live cells on the surface of your skin, the surface of the skin is made up of dead cells. Here is a neat diagram given here. So now, this book covers about a cut that has happened in your toe, and many millions of bacteria have entered. So what happens is, the first line of defense, the macrophages come in, and then what happens is they swallow the bacteria whole. As discussed in the previous, previously, these macrophages are big, so they can swallow the bacteria whole. Then they order neutrophils to come, which order inflammation. Inflammation releases fluids into the infected area, which carry many antibodies and warmth, which stops the bacteria from growing. Then these neutrophils release acids and even release their genetic material to form a net kind of things. There is a Then what happens is this. Then there is dendritic cells that come in and then take samples from these bacteria. Now what happens is they just talk about the what do you say, phagocytosis. They talk about phagocytosis, which means phagocytosis means the eating of a bacteria for a cell. Like anything in the single cellular world, a bacteria comes, is divided by the macrophage, it is broken down by acids and then released. Now they now tell us about inflammation. Inflammation releases heat. It releases heat. What happens is bacteria cannot function in the heat, which that is why if you get a cut and it is inflamed, it is warm. That is why. Now they tell you about certain proteins. So how do cells communicate? Like, how do cells communicate? Like, let's say, if how does macrophage allow a neutrophil to come? It is all because of cytokines. They are special proteins which make the cell know where to come and what to do. They have special receptors for cytokines, and they can tell very small differences in concentration of cytokines and can detect where the cytokines are coming from. Here is a neat diagram given. They are now telling about receptors. Receptors are just special things. Let's talk them about puzzle building blocks which connect from one to another, like given here. So this special receptor will only catch this protein, connect to this special protein which looks like a W. This receptor cannot get this one, but this receptor can get this kind of special protein. Now they talk about the complement system. So complement system is a special kind of proteins that make the bacteria unable to do anything and kill them. There are 30 kinds of complement proteins, as you can see here. Now, what happens here is this. First, a C3 comes. And then the C3 breaks into C3b and C3a. The C3a then goes to the immune system to activate the macrophages and neutrophils. Then the C3a converts a B protein comes in, and it just splits, and in the end it finally converts into C3 convertase, which activates more C3 proteins, which then now what happens are like now they just like convert them into C3. Now special complement proteins come in and make a hole in the bacteria, which makes the body fluids rush in outwards and the bacteria is dead. So this is how the complement system works. Now remember what happened with the samples that were collected by the dendritic cells. They go to special cells called T cells. T cells are cells which recognize this special kind of what do you say, the sample. This sample here is called an antigen, just for your. So each T cell responds to a different antigen. Let's say there is an E. coli sensing T cell, E. coli antigen sensing T cell, and the bacteria that comes is like not E. coli, so they will not. The dendritic cell will not activate the T cell. So now let's say if an E. coli antigen dendritic cell comes into contact with an E. coli sensing T cell, it would then activate it, and then the T cell will start to replicate. As given in here, they collect samples. Now we'll talk about where these T cells go in the higher chapters, but first let's talk about the lymph nodes. So lymph nodes are special tubes that connect every part of your body. They also collect dead cells, which is why sometimes there is a yellow thing on the back of your throat. Here is a diagram given for the lymph node, lymphatic system. Now these T cells, which were activated by the dendritic cell, now turn into helper T cells. Now these helper T cells divide into two groups. One goes to B cells. We will talk about that later, but the other groups go to the macrophages and neutrophils fighting on the infectious area. But first things first, macrophages can only devour 100 bacteria before they die. So if a special helper T cell connects to the macrophage, it can allow the macrophage to work harder and devour more bacteria. So now we come to the adaptive immune system. So now what happens here is this. The T cells activate B cells, which in turn release antibodies. So now there is a question. There are every possible There are every possible antibody sequence for every bacteria. Let's say there is a bacteria that emerged on, let's say, Mars in 1000 years. Your immune cells have that kind of antibody. So how does this happen? So what happens is the B cells are trained to make certain antibodies. It's like mixing two things to find new things. It's like that. Let's say you have 5 things. You need to mix them in every possible way. You mix them in every possible way to make every possible antibody. It's like that. But would the antibodies actually harm your immune cells? Because there is a certain case in which the antibody attacks the immune cells. Yes, there is. That is why some people get autoimmune diseases. But if the teacher cells in the bone marrow and the thymus, which we will talk about later, detects that it releases antibodies that could harm the immune cells or other cells in our body, it destructs the cell. So yeah. Now they talk about the thymus. Here is the thymus. As I have said earlier, the thymus trains T cells. Now these T cells, what happens is this: the T cells attach to the dendritic cells, but sometimes the T cells can activate on their own. Because so what happens? They go through a training where the same thing happens with the B cells that I have talked earlier. It's about mixing and matching. So so this training happens. There are 3 tests. So if it can release cytokines, it passes the test. Or if it doesn't, then it dies. And if it there are three tests given for the T cells. The first is like if they have functioning receptors. The second is if they are good at communicating or releasing cytokines, and if they can recognize self. Meaning if they can recognize self. If they can recognize self, they are immediately destructed, because if they can recognize other cells of your own body, it has a right to destruct them, which causes harm to your own body. That's why it is destructed. Now they just talk about antigens, which I have talked earlier. Now they talk about the activating T cells. Now they activate by two cells step verification. They first get into the first receptor. They first like detect the MHC class 2 molecule, and then they just connect with the receptors of the dendritic cell. That's how they get activated. Now, the B cells have a similar training. They go through bone marrow training, and if they get activated through antigens, which means that if the T cells activate the B cells, they release a short number of antibodies. But then after they also the lymph also contains dead cells, which can have like dead viruses or bacteria. If they sense that, they then turn into plasma cells. Now these plasma cells then convert into long-lived plasma cells, which then create, if the infection happens again, they just release small amount of the same antibody. And the B cells, after the infection is over, they turn into memory B cells. This is how things work. If the antigens float through the lymph node, they get activated. Now the B cells are activated. Now the the dendritic cell activates the T cell, which activates the B cell, which converts them into plasma cells. Now these talk about antibodies. Now there are like 5 classes of antibodies: IgM, IgE, IgA, IgE, and IgG. There are many so they only talk about four antibodies, because IgD in a nutshell doesn't do anything. So like you can see, they're literally pincers. They're literally like crab shells with like pincers on it. Now they talk about mucosa. What is mucosa? What happens is this: the inside linings of your organs is called like mucosa. It is covered with mucus, the same stuff which comes out of your nose. So mucus has a lot of different antibodies, and the gut mucosa has a lot of different microbes, and there is only an epithelial layer of one cell, so they can easily venture out. So it is necessary to have a mucus layer and antibody, and there are many immune cells there too, to devour the bacteria if they go too deep. Here is the gut mucosa, the epithelial cells, and the macrophages, the B cells, the helper T cells, and these are the M cells. These are the commensal bacteria, which are good bacteria for you. They help digest things that are not digested by you. So yeah, now they talk about viruses. What is a virus? Virus, as you can see here, has protein, is literally a protein with a protein they are literally DNA with a protein layer. Basically they are all proteins. They are the distinction between dead and alive. Now they talk about the infection of the influenza virus. So the virus connects to the receptor cell in your cell, then it enters the cell, then it goes to the nucleus and orders more of its own kind to be replicated. Then what happens is this, then they just destroy the bacteria. So now if they do this, they wouldn't let the immune system know. So how does the immune system know? So the cells which are then infected by the virus release some special chemicals called interferons. They interfere with the virus and help to communicate with the other cells to slow their production down. So, and what happens if then there are natural killer cells? They come in, and what happens if they detect a infected cell, they order it to kill, and then macrophages eat the leftover thing. That's how influenza is dealt in your body. I hope you enjoyed. See you later. Bye.